Organic compounds, such as glucose, store energy in their arrangements of atoms. These molecules are broken down and their energy extracted in cellular respiration. The first stage of cellular respiration occurs in the cytosol, while the second and third stages occur in mitochondria. In cellular respiration, electrons are transferred from glucose to coenzymes such as NAD+, and finally to oxygen. The energy released by this relocation of electrons is used to make ATP. Carbon dioxide and water are given off as byproducts. Click on each stage of cellular respiration for a brief description. Glycolysis is a series of steps in which a glucose molecule is broken down into two molecules of pyruvate. As the chemical bonds in glucose are broken, electrons and hydrogen ions are picked up by NAD+, forming NADH. Glucose is oxidized and NAD+ is reduced. A net output of 2 ATP molecules are also produced in glycolysis for every glucose molecule processed. But most of the energy released by the breakdown of glucose is carried by the electrons attached to NADH.
The pyruvate molecules are modified as they enter the mitochondrion, releasing carbon dioxide. The altered molecules enter a series of reactions called the citric acid cycle. More carbon dioxide is released as the citric acid cycle completes the oxidation of glucose. 2 ATPs are formed per glucose, but most of the energy released by the oxidation of glucose is carried by NADH and FADH2. Almost all of the ATP produced by cellular respiration is banked in the final phase, oxidative phosphorylation. The NADH and FADH2 molecules produced in glycolysis and the citric acid cycle donate their electrons to the electron transport chain. At the end of the chain, oxygen exerts a strong pull on the electrons and combines with them and hydrogen ions, protons, to form water. The electron transport chain converts chemical energy of moving electrons to a form that can be used to drive oxidative phosphorylation, which produces about 34 ATP molecules for each glucose molecule consumed.
Glycolysis, which begins the breakdown of glucose, is a series of 10 enzyme-catalyzed chemical reactions that can be divided into two main phases. In the energy investment phase, some ATP energy is used to start the process of glucose oxidation. By the end of this phase, a 6-carbon molecule, glucose, has been split into 2 3-carbon molecules of glyceraldehyde phosphate. The 3-carbon glyceraldehyde phosphate molecules now enter the energy payoff phase. Chemical bonds are broken, and NAD+ picks up electrons and hydrogen ions, forming NADH. The energy released is used to attach phosphate groups. The phosphates are transferred to ADP, finally making some ATP. This way of making ATP is called substrate-level phosphorylation. A couple more reactions rearrange the atoms in the 3-carbon molecules. More ATP is generated in the final reaction that yields pyruvate. For each glucose molecule broken down during glycolysis, a net of 2 ATPs are formed, along with 2 NADH molecules.
The oxidation of glucose continues in the citric acid cycle. Pyruvate molecules formed during glycolysis diffuse from the cytosol into the mitochondrion, but pyruvate itself does not enter the citric acid cycle. A reaction occurs that removes a carbon atom, releasing it in carbon dioxide. Electrons are transferred to an NADH molecule, storing energy. Coenzyme A, or CoA, joins with the 2-carbon fragment, forming acetyl-CoA. One molecule of acetyl-CoA enters the citric acid cycle. The 2-carbon fragment of acetyl-CoA attaches to the 4-carbon molecule, oxaloacetate in the first reaction of the cycle. This forms citrate. In a series of steps, bonds break and reform. 2 carbon atoms are released one at a time in molecules of carbon dioxide. Electrons are carried off by molecules of NADH and FADH2. One step produces an ATP molecule by substrate-level phosphorylation. A 4-carbon oxaloacetate molecule is regenerated. Since 2 acetyl-CoA molecules are produced for each glucose molecule broken down, a second acetyl-CoA enters the citric acid cycle. The same series of reactions occurs, releasing carbon dioxide and producing more NADH, FADH2, and ATP. The cell has gained 2 ATPs that can be used directly. However, most of the energy originally contained in the bonds of glucose is now carried by the NADH and FADH2 molecules. Most of the energy harvested from organic molecules during glycolysis and the citric acid cycle is stored in NADH and FADH2. These molecules give up their high-energy electrons in the third phase of cellular respiration and oxidative phosphorylation, where most of the cell's ATP fuel is produced.
The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondrion. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward highly electronegative oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of electrons to an oxygen atom and 2 hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed-up hydrogen ions give up their energy when they diffuse through a special protein in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP. This mode of ATP production is called oxidative phosphorylation because it is powered by the transfer of electrons to oxygen. Under normal conditions, almost all the ATP produced in the process of cellular respiration is manufactured by electron transport and oxidative phosphorylation, about 34 ATPs for every glucose consumed.
This illustration summarizes cellular respiration. Glycolysis and the citric acid cycle produce a small amount of ATP via substrate-level phosphorylation, but most of the cell's ATP is made via oxidative phosphorylation, when NADH and FADH2 produced in glycolysis and the citric acid cycle give up to oxygen the electrons obtained from organic molecules.